Within the 3-pound universe of the human brain, a cosmos of thought, memory, and identity unfolds every second. It is the most complex structure we know, a delicate network of 100 billion neurons firing in silent, perfect harmony. But within this sanctuary of the self, a silent storm can gather. A disease so aggressive, so relentless, that it challenges the very foundations of modern medicine. This is glioblastoma, not an invader from outside, but a rebellion from within. Understanding it means confronting the brain's profound fragility and the remarkable resilience of human life. Glioblastoma, classified by the World Health Organization as a grade 4 astrocytoma, is the most common and most aggressive primary brain tumor in adults. It doesn't arise from the neurons that carry our thoughts, but from the cells meant to protect them. Glial cells, specifically astrocytes. These support cells are the brain's housekeepers and guardians, providing nutrients, maintaining chemical balance, and insulating delicate neural circuits. But when the genetic instructions within an astrocyte catastrophically break down, this guardian cell turns traitor. It begins to divide uncontrollably, its progeny forming a chaotic, disorganized mass. The grade 4 designation is a measure of its ferocity. It signifies rapid growth, a tendency to outgrow its own blood supply, leaving a core of dead or necrotic tissue, and an almost unstoppable drive to expand. What makes glioblastoma so formidable? First, it defies a surgeon's scalpel. Unlike many tumors that grow as a contained spherical mass, glioblastoma is infiltrative. It doesn't form neat borders, it sends out microscopic finger-like projections that weave themselves into the intricate fabric of healthy brain tissue, like roots spreading through soil. This means surgeons can't just cut it out cleanly. Removing every cancerous cell is a near impossible task without damaging critical areas responsible for speech, movement, or memory. The surgeon's goal becomes maximal safe resection, removing as much of the tumor as possible while preserving the person. But inevitably, rogue cells are left behind. Second, the brain's own fortress works against us. The blood-brain barrier, a highly selective membrane that protects the brain from toxins, also blocks many life-saving chemotherapy drugs from reaching the tumor. It's a brilliant defense mechanism that, in this case, shields the enemy. Third, the tumor is a master of disguise and adaptation. It is not a single uniform entity, it's a chaotic mix of cells with different mutations and vulnerabilities, a concept known as intratumoral heterogeneity. This genetic instability is its superpower. When a therapy attacks one type of cell, it often leaves behind resistant survivors. These survivor cells then multiply, creating a new, more resilient tumor that no longer responds to the original treatment. Therapies often leave survivors that regrow, stronger than before. Because the brain is the seat of our entire being, the symptoms of glioblastoma are deeply personal and depend on where the tumor is located. In the frontal lobe, patients may show personality changes or difficulty with planning. In the temporal lobe, it can cause seizures or memory problems. A tumor in the parietal lobe may lead to weakness or sensory loss, while one in the occipital lobe can impair vision. The most common first symptom is a new, persistent headache. Unlike prior headaches, often a dull, constant pressure that worsens over time. Other signs can be subtle or sudden: seizures, a new clumsiness or weakness on one side of the body, trouble finding words or understanding speech. For the patient and their family, it can feel like the person they know is slowly being erased or altered, as the tumor disrupts the very neural circuits that define them. Even with our best treatments, the prognosis remains daunting, with a median survival of about 12 to 18 months. The diagnostic journey typically begins when these symptoms prompt a visit to the doctor. The key tool is magnetic resonance imaging, or MRI, with contrast dye. On these scans, glioblastoma often appears as a characteristic ring-enhancing lesion, a bright, irregular ring of active tumor cells surrounding a dark necrotic core, where the tumor has grown so fast it has outstripped its blood supply. To be certain, a neurosurgeon performs a biopsy, taking a small tissue sample for a pathologist to examine under a microscope. Today, diagnosis goes beyond imaging. Molecular testing is essential. These tests read the tumor's genetic profile and inform prognosis and therapy. Most adult glioblastomas are IDH wild type, which generally behave more aggressively than IDH mutant tumors. Another key marker is MGMT promoter methylation. When the MGMT promoter is methylated, effectively silencing the gene, the tumor is less able to repair the DNA damage caused by the standard chemotherapy, temozolomide, and patients are more likely to benefit. Other frequent alterations include EGFR amplification and TERT promoter mutations, each contributing to the tumor's relentless growth. With this information, a multidisciplinary team, neuro-oncologists, neurosurgeons, and radiation oncologists, creates a plan. The current standard of care, called the STUP protocol, uses 3 coordinated steps. First is maximal safe surgical resection to remove as much tumor as possible. Next comes 6 weeks of focused radiation, delivered 5 days a week, to target remaining cancer cells. During radiation, the patient takes daily oral temozolomide. Afterward, higher dose adjuvant temozolomide continues for several months. In recent years, a 4th modality has been added to this regimen for some patients: tumor-treating fields. This involves wearing a portable device with electrodes on the scalp that generate low-intensity electrical fields. These fields are designed to disrupt the process of cell division, specifically targeting the rapidly dividing cancer cells, while sparing healthy, non-dividing brain cells. For some patients, this has been shown to modestly extend survival when added to standard chemotherapy. Despite this aggressive multimodal approach, recurrence is, unfortunately, the expected course. The tumor's heterogeneity and infiltrative nature mean that resistant cells almost always survive and begin to grow again. When this happens, options become more limited. Clinical trials testing novel therapies are often the best path forward. In select cases, additional surgery or re-irradiation may be considered. Other chemotherapy agents, like lomustine, may be used. Another drug, bevacizumab, can be helpful. It doesn't target the tumor cells directly, but instead blocks the formation of new blood vessels, which can reduce brain swelling and alleviate symptoms, improving quality of life even if it doesn't dramatically extend it. So, what's next? Where's the hope? It rests in relentless science. Researchers are testing a range of innovative strategies. Immunotherapies aim to mobilize the patient's own immune system against the tumor. These include personalized vaccines made from tumor material and dendritic cell vaccines that train immune cells to recognize cancer. Genetically engineered oncolytic viruses are designed to selectively infect and kill tumor cells. CAR T-cell therapies, successful in blood cancers, are being adapted for glioblastoma, with engineered T-cells targeting proteins such as EGFR-V3 and IL-13R alpha-2. A common question is, who is at risk? For the vast majority of patients, glioblastoma is sporadic, meaning it occurs by chance with no clear cause. There are no established lifestyle preventions, and despite persistent myths, large-scale studies have found no conclusive link between cell phone use and glioblastoma. However, a few known risk factors exist. Prior high-dose radiation to the head, often for a childhood cancer, is a definite risk factor, though this is very rare. And certain inherited genetic syndromes such as Lee-Fraumey-Nice syndrome and neurofibromatosis type 1, or NF1, significantly increase a person's risk of developing brain tumors, including glioblastoma. In the face of such a difficult diagnosis, it's easy to get lost in the statistics, but the hard truth is that while numbers matter, people matter more. Certain factors are associated with better outcomes: younger age at diagnosis, good functional status, and that favorable MGMT methylation biomarker. But beyond the biology, the human elements are just as critical. Quality of life, aggressive symptom control, and a strong support system of family, friends, and caregivers are paramount. If this disease touches your life, or that of someone you love, the most important step is to seek care at a comprehensive cancer center with a dedicated neuro-oncology program. Ask about multidisciplinary care and available clinical trials. Knowledge isn't a cure, but it is a plan, the foundation for informed decisions, finding the best possible care, and holding on to hope against one of nature's toughest challenges.